This program is brought to you by Stanford University. Please visit us at stanford.edu.
Let's do a little quick review. So the first thing we did in the class was flow sheets and units. Parries. And the flow sheets took us into what concept? Mass balances. So someone say the mass balance mantra. I'll start it with what goes in
must come out or accumulate.
Minus what comes out equals accumulation plus reaction. Okay? So that took us through mass balances. And we learned to take a complex process. When you make mass balances, you always draw the little control volume, and you make the balance across anything that's crossing that control volume, except if something's going on inside the control volume, such as reaction. You need to take that into account, because you can either be consuming or producing a component. We also have what kind of, you have units and they're back connected, recycle stream. So you learned how to handle recycle stream. And then after we talked about those mass balances, that led us into what? Energy balances. And so what's the mantra for energy balances? Energy in minus energy out equals accumulation of energy, which can be also depletion, right? Depends on the sign. Plus the energy that could either be consumed or produced by reaction. So it looks just like the mass balance, except it's energy.
You also learned how vampires work.
You had what?
You learned how vampires work.
Okay, so then, I'm trying to think what came. Then after that, after we did the energy balances, what did we talk about next? Okay, we did heat transfer, and in the context of heat transfer, what did you learn how to design? What? Heat exchangers. And we found out they could be outfitted in one of two ways, co-current and counter-current. And why is one more efficient than the other?
Minimize temperature difference.
You take better advantage of the temperature difference between the two streams by running co-current rather than counter-current, rather than having this huge temperature differential at the beginning when you're running co-current and sort of wiping it out slowly, making the back end of the device pretty inefficient. So we found out that counter-current is always more efficient than co-current. And then people ask me, why do we even talk about co-current? Why? Well, if I didn't, you might go design one. And so now you know not to do that. Then after we did heat exchangers, what did we jump into? Scaling. And what concepts did you learn in scaling? Buckingham pi theorem, which allows you to do what? Find
out that giant mice can't walk.
That's true. Giant mice can't walk. Well, they can for the first step. And so there you have a complex, could be a very complex problem, and you ask yourselves, what's the minimum number of dimensionless groups that I need to describe that problem? And that's what Buckingham pi gives you. So it reduces essentially the experimental challenge you have ahead of you in trying to understand how all those variables relate to one another. And we did the walking problem, where we saw that we could actually reduce it to two non-dimensional variables and describe how even four-legged and two-legged things propel themselves at what speed based on their stride length and their hip to foot length, which I thought was pretty good. Then we also looked at in getting into the problem of pharmacokinetics. And using ways of thinking about how to reduce, again, a complex set of data. Remember the data on the methotrexate on all the animals that was all over the place? And with a really simple analysis based on scaling arguments having to do with how body weight and organ volume and metabolism scale, we were able to reduce that to one curve. And I told you that was a really important kind of finding in the 1970s with regard to animal modeling with drug discovery. And then after we departed from there, where'd we go?
And moving human with.
Right. So I used the human thing, but really what we were doing is doing mass balances again, but with what additional? Non-steady state. So unsteady state mass balances. And we started out with 2 tanks interacting. And then we, on Friday, give Max a hand. It was hard work. It's getting wimpier every time, Max. I mean, you know, they're losing their energy. Designed a multi-tank model to represent the human body. And that's this computer project that you're working on. So now what we're going to do is I'll spend a couple lectures on roughly the topic is biomedical engineering, how chemical engineers apply some of these concepts to solve important problems in medicine. And so the next 3 lectures, including this one, are going to revolve around how the human kidney works. And if you understand how the human kidney works, then you'll be in a position to design an artificial kidney. And we'll do that, and then we'll go over to the Veterans Administration Hospital and see patients hooked up to artificial kidneys. And that's on Monday, is it? Next Monday, a week from, is it a week from today? You're sure? Okay, he's sure. Anyway, it's in your syllabus. Now that's again another bike trip. And it's a little longer, but not much. It's around the corner from the blood bank. Okay? So if you get enough speed, you can actually coast from the blood bank to the VA. And Tim Meyer, who runs the nephrology clinic at the Veterans Hospital and also for Stanford, will be our host. And I just have to warn you, he really gets into this tour. And I don't want him to frighten you. He's not being aggressive. He's just into wanting to teach about how artificial kidneys work. And you might have a hard time understanding how someone could get so excited about this, but so don't antagonize him. In the what department?
In the bio department offices in Gilbert.
No, that's a different one. This Tim Meyer is, I've known him for 30 years, and he's, and we've been doing this tour for almost that many years, and the machines have taken dramatic changes from the first tour to now. I'll explain some of those concepts to you. So I'm going to tell you a story, and then we'll do a little physiology today, because you need to know what the kidney is all about and how it works. So when I came here to Stanford in 1969, as I told you before, the Vietnam War was raging, and I didn't want to participate. And you know, I'd moved to Europe and then gotten some kind of an exemption to come back to this country and work in a critical industry. And that's when I went to work for the Marathon Oil Company. That got me the exemption. And then, you know, I got fired in 3 weeks, but I didn't report that to the draft board. For all I know, they thought I'm still working there, although we don't have draft boards anymore. And I came to Stanford, and I was asked to start a program in bioengineering. Or biomedical engineering. No one really knew quite what that was, but I did know one thing. I didn't want to come back to the department where I had received my PhD and compete against my advisor, like do the same thing I had been doing, because I'd just be a tool. And so, I don't want to be like Professor Tool. And so I didn't know what I was going to work on. And 2 graduate students picked me to be their advisor for their PhD. So I'm 26 years old and they're 22. So they're almost essentially the same age. So we became great friends. And both of them were conscientious objectors. So they didn't want to go to the war either, and this is why they gravitated to me. Now, I was also sort of playing the part. I had hair down to my waist. I'd wear tank tops and Bermuda shorts and thongs to lecture in. I mean, it was sort of like the cool thing to do. And they looked the same, so we were indistinguishable. And so Bill Dean, who's now a very a well-known professor at MIT in the chemical engineering department. In fact, has written one of the best-selling books in transport phenomena, which is fluid mechanics, mass transfer, and heat transfer, was one of my first students. The other, Bob Bradshaw, who works at Sandia Labs in Livermore, he and I still ski together every year. And the 3 of us used to go on hikes in the Sierra called Old John hikes after John Muir. And we'd go up on Friday night and we'd bag 2 13,000-foot peaks, one on Saturday and one on Sunday, and then drive back. And I'd go back to class on Monday, dead tired as you might. And these were pretty awesome climbs. I mean, if we had packs on our backs and if they got too heavy, these guys would just take their packs and chuck them off the mountain and just just get rid of them, get rid of the weight, chuck your backpack and your sleeping bag and everything. It was good. It was the Old John trip. And so Bill Dean says, I need to work to keep my conscientious objector. I have to work on some healthcare problem that's important. So he and I actually went over to the medical school and started knocking on doors, saying, Channing and Bill are here to help you. And we ran into a guy. It's like selling Amway. Slam, and you walk away and you go in the next door. And finally, this older prof, Roy Maffley, said, you know what? You guys are too ahead of your game. Stanford's too conservative to handle you people. But I know a guy who teaches at UCSF, University of California, San Francisco. He's just starting out, young MD in nephrology. And he's been taking some really interesting data. And you ought to go talk to him. And so we went and talked to him. And in the next lecture, I'll tell you what he told us and the data that he gave us. But as you might imagine, we go up there cold turkey talking to a nephrologist, and he starts talking about the experiments he's doing on kidneys. And all I knew at that point is we had 2 of them. And so we were starting at a huge disadvantage. So I'll help you overcome that disadvantage by handling a little bit of how the mammalian kidney works, because Bill and I had to come back and figure this out for ourselves, and then try to take his data sets and animal work and fold them into what was known. And I just give you a little end of the story. The 3 papers that we wrote in 1971 describing how the mammalian kidney processes blood are now taught to every medical student in the world. And so I'm not taking credit for it. Bill really did the work, the graduate student. But a simple mathematical model, which I will go through next lecture. We'll do the whole model in 45 minutes. Change the way people think about how the mammalian kidney works. Up until then, it had just been, well, blood in, blood out, and some urine, and it's cool. And if no urine comes, you've got a medical problem. It's sort of like that. And so give you a pill. And then if the urine doesn't come, you finally blow up. And so then it's over, and then you don't need any more pills. And it's kind of like, okay, that's the way it worked. So we fixed that. So let's start by, I've already given away part of it. You have 2 kidneys. What's that?
I haven't heard that story before. Did you make it up?
Which one? Which story?
Never mind.
There aren't any stories in this class. It's all truth in advertising. So you have humans have 2 kidneys. All mammals have 2 kidneys. Ours are about the size of our fist. They reside more toward the back than they do the front. If any of you saw that exposition called Body World that came through San Jose not too long ago, where this guy plasticizes humans, you can see the anatomy really pretty plainly. If you didn't see that, you can imagine this is what it would look like. You have the heart up here pumping blood down the aorta, which is a tube about the size of your fourth finger, maybe your middle finger, and some people even larger. Comes down over the arch, comes down, splits into the iliac artery, goes down your legs. There is an offshoot of the aorta called the renal artery. There's two. One goes to your left kidney, and the other goes to the right kidney. That's called the renal artery. So that's one piece of plumbing into the kidney, the blood in through the renal artery. The blood has to come out after it's processed, and it comes out the renal vein. There's two of them, and they get dumped into what's called the inferior vena cava, or cava, which is a very large vein that's returning blood from the legs back to your heart. And they basically runs parallel to the aorta. Now, there's a third tube that comes out of the kidney. It's called the ureter. And so that third tube is containing the waste materials that have been removed from the blood. And that, in a very simple, is what you're looking at in terms of plumbing. Now, the reason that Bill and I found the kidney an interesting thing to think about is it's not connected to your mind. Now, how do you know that? Well, you know it's not connected to your mind because you know you can take a kidney out of one person and put it into another, and it will work. And you're not plugging it into the brain, where the brain is somehow controlling it. You're just hooking up these three tubes, and off it goes. And so this seemed like a really good organ. The heart's more complicated because it's got, all these electrical signals in it and so forth that need to be, modeled if you're going to model the heart. So this seemed like a good unit operation, if you will. It's got flow in and two flows out. And then the question is, given the contents of the material going into the kidney and the contents in the ureter and the contents coming back out and the flow rates and the pressures, how do you explain how this thing works? Under situations where it's compromised, maybe you have high blood pressure, maybe you have low blood pressure, maybe you have something wrong with the little filtering units in here and so forth. How can you detect that, and how can you think about therapies to overcome deficits in kidney function? So with that little piece of background, let me just go to the board for a second and draw some pictures. So we're talking about renal physiology. And sort of the exercise I'm taking you through is when someone gives you a complicated problem about something you know nothing about, find out what it is you're supposed to know before you start thinking about the problem. So our thing was find out something about what's known about the kidney. Now, first of all, the cardiac output, which is coming out of the heart. Do you remember what that is typically in an average human? How many mLs per minute? 5,000. So it's roughly about 5,000 mLs per minute. And then the next question you might ask is how much of that blood goes to the kidneys? Obviously, some fraction of that does. Well, interestingly enough, about 25% of the blood, or approximately 1,250 mLs per minute, get processed by the kidney. So a quarter of what your heart pumps goes to the kidney. Now, what is the kidney doing with the blood? Why does the blood go there? Does it go there just because it's going for the ride? It what?
Filters.
It gets filtered somehow. And why does it get filtered? What is the kidney trying to accomplish?
Get rid of waste.
Get rid of waste. Can you think of what the wastes are that the kidney gets rid of?
Ammonia.
Nitrogenous metabolites, such as urea. That's one. Creatinine. I can't spell it. T-I-N, creatinine. Is this right? Yeah, it looks about right. And uric acid are the primary ones. These are basically the nitrogenous metabolites that flow from the destruction of protein in your body, which has nitrogen in it. So these are all containing nitrogen. And that's primarily what the kidney is doing, is monitoring that. And it controls also something else. Remember I told you the story about the guy drinking Sterno and the baby blowing off CO2 and was crying? What was being controlled there? You don't remember that story, do you? That's because it was, what, 2 weeks ago? So pH, it controls hydrogen ion concentration, the kidneys do. And it controls one other thing. I'll give you a hint. You're at sea, bobbing in a rowboat between Fiji and the Bering Straits, and all you have to drink is seawater. Salt, okay, or sodium chloride. So it's a big, and to some extent, potassium chloride. Or you can think of it as sodium, potassium, and chloride ion. It's a very important modulator of salt. And that's really important because, as some of you who've had biology know, that, and to some extent calcium, these are really very important in terms of their distribution across cell walls. And if you get that upset, you can get yourself very sick. Now, this material, and by the way, if the hematocrit is about 50%, that is, your blood's 50% by volume red cells, what does this translate into in terms of plasma flow? This is blood flow. So what would the mLs per minute of plasma flow be to the kidney? If your hematocrit's 50%. How much?
625.
Yeah, 625. These aren't trick questions. Of plasma. And why do you think I'm focusing on the plasma flow rate?
That's where this stuff is dissolved?
That's where it's dissolved, and that's what gets filtered. You see, there's no use to filter red cells. So as the blood flows into the kidney, you want to keep the red cells on the blood side and filter the plasma. Now, so then the next question is of the 625 mLs per minute of plasma that are entering the two kidneys, how many mLs per minute of this actually ends up in the filtrate? That is, passes out of the capillary systems and gets collected and processed. And it turns out to be 125 ml a minute. So of this 625, 125 gets filtered and processed, which is about another 20% of the plasma. So you can think about it, 25% of the plasma gets filtered, and 20% of what gets filtered goes to the kidneys, 20% of that gets filtered and gets processed. Now, how much urine are you making? Anybody have any idea how much urine you're making right now sitting there, provided you didn't have a six-pack for lunch? So it lets you out, Red. Theo, you're out. Michelle, you're out. Who else is out? Come on, admit it. I'm out. You make about 1 ml a minute of urine. So if you have the kidney, and you're putting in 625 mLs per minute of plasma, and you're filtering 125 mLs per minute of that plasma, and you've got another stream coming out here, which is 1 ml per minute. What does that say? It must be happening inside the kidney and coming out here.
Stuff is reabsorbed. Reabsorbed.
So first of all, you've got to reabsorb how much?
124.
124 mLs per minute. Add it back to what short-circuited here to give you how much coming out. How much plasma is coming out here? Remember, mass balances, people? This is not rocket science. 624 mLs a minute. Really interesting, isn't it? You've got this organ, and it's taking in a lot of fluid, and it's putting almost all of it back, minus this little measly bit of 1 ml per minute.
Where does the system pick up the liquid again that it lost? Like it just in the bottom?
It just short circuits through, it never gets filtered.
No, like where does it pick up like the extra milliliter? Like you drink water, right? Where does
Oh,
in
yeah.
the body in general, where does it pick up the liquid that you?
This 1 ml,
Yeah.
when you drink the 6-pack,
Where does that happen? Like
went through your mouth.
Is there any intestine? Like in the like just
Yeah.
Okay.
Yep, yeah,
Serious.
yeah, no. Stomach, you know. Anywhere you can leak across,
Okay.
it'll get in there. Yeah, so, you know, you're out in the desert, there's no water, you keep doing this, and then finally, what happens? You start to get tiny, right? Because you're losing, and you're evaporating fluid and so forth, and then you finally die of thirst. You notice what's going on here. If you have a lot to drink, a lot of fluid, what color does, if you happen to be looking, does your urine become? Clear. And if you don't drink a lot for a while and you go, it's really golden or bright yellow. And all you're doing is looking at the concentration of solutes in there that absorb in the visible spectrum most of these nitrogenous compounds. If you want to get an idea of how fast your kidney is working, all you have to do is eat some asparagus and wait about 10 minutes if you need to pee, and what? You smell it. I mean, it's really fast. I mean, it's a little experiment. You can do that. You can eat the asparagus and then wait 20 minutes, and then eat the asparagus and then 15 minutes. You could plot, the sniff test, and you can see how fast it goes.
Non-dimensionalize it.
And then Estelle says, non-dimensionalize it. That's right. So now, how does this happen? This happens in a series of capillary networks. So let's put this is pictorial. Here's the renal artery coming in. And this bifurcates many times, and I'll just show two, into arterioles. So this big vessel keeps dividing. Inside the kidney. And let's just follow this particular one. And this keeps dividing again into capillaries. So the difference between an artery and an arteriole and a capillary is just the diameter. These capillaries can be, let's say, they can be as small as about 6 microns in diameter. They're really tiny. Red cells are about 8 microns. And so they're getting kind of squeezed down now as they get through these. But what's happening to the surface area of all the tubes? It's going up hugely. Every time you do this, you can think of it as n to the 2, or 2 to the n. And you finally get down to the capillary bed, which looks like a cobweb of very, very small vessels, all being fed here from the renal artery. And then, of course, what happens? These capillaries come back and join together, and then others come in and join together, and you have a return flow finally out the renal vein. So the picture you should have in your mind is the blood's going into this artery, splitting into arterioles. They split into capillaries. You have this big capillary bed. Then it all reforms into arterioles and kind of goes out the renal vein. And so the kidney is, in fact, a mass of capillaries. And it's across these capillaries that filtration of plasma occurs and reabsorption occurs. And the red cells, they just merrily go on through. They're big and they're held back. So this is a very sophisticated filtration mechanism. The reason, of course, we need to know this is we have to have some understanding how the kidney works, so that if we design an artificial kidney, we at least perform some of the functions that the real kidney does. Now, the next question is, how, compared to other organs, how does the kidney behave? Which is sort of interesting how nature has optimized this system and made it. So you'll see the kidneys are getting most of the blood flow compared to, and this is milliliters per 100 grams per minute. So this is normalized to the weight of the organ. So it's really very, very high for the kidney, more so than any other, more than your brain, your skin, and skeletal muscle, and so forth. So that's, I think, an interesting observation. And by the way, the kidneys are so important to you. How is it that I can donate a kidney to one of you? If both your kidneys shut down, you have to go into a dialysis machine or you'll die, unless someone gives you a kidney. Now I'm just left with one. And it turns out it adapts and works just fine. You don't actually get any more surface area, you don't get any more filtering. Isn't that an interesting sort of redundancy that nature has built in? In other words, now you don't have that same redundancy if you lose half your brain. And it's probably because losing half your brain is tougher than maybe getting a gunshot or back in the Middle Ages, a spear or something through your kidney. If you get a spear through your head, then it doesn't matter what they do to your kidney. You're walking around, like this, and you're brain dead.
Partially dead because it wastes so much. Like if 5,000 flows in and only 1,250 gets actually filtered, does that extra capacity get used?
Well, 1,250 is the plasma that flows in.
Okay. Oh yeah.
Right, and 125 is how much is being filtered.
So does your kidney get larger?
No, actually it doesn't. What it means is that you have a processing capability of a dynamic range of an ability to process these solutes in a way that if you were to lose half of it, the other one can make up for it by changes in both pressures and flows to that remaining kidney. And that's how it adjusts, because it's not connected to your head. Your head's not telling it, oh, the blood's getting dirty, work harder. Okay, so there's a compensation that goes on. It's very interesting. Glomerulonephritis is a disease that strikes people. And the little filtering units in your kidney, called glomeruli, which we'll learn about, if you lose them in disease, you've lost them forever. You don't make any new ones. So the kidney is one of the organs that does not rejuvenate itself. The liver does. If you hack up part of your liver will actually grow back, but not the kidneys. Now, what I didn't point out is where this extra tube goes. It goes down to a holding tank. Did you have a question, Michelle?
Yeah, so where does the 500, like, how does the 500 mils that doesn't get filtered, like, short circuit or work?
It just stays in the capillary. You see, if you were to filter all,
So are there more like branches that go off, and then that's where the filtering happens, and then it comes back?
you can't filter 100% of the plasma that comes into this capillary bed. If you did, there'd be nothing left in the capillaries and they'd collapse. So you filter just a fraction of the plasma that's coming in, roughly about 20%. And the rest is used to carry the red cells through the system. Can you think about that? If you have this suspension coming in, and if I were to filter it all out and leave the red cells behind, I'd have muck. You'd have oatmeal, and everything would be clogged up. So you can only filter, and you can imagine if I'm filtering out a lot of that plasma, what happens to the red cells? They find themselves in a less of a fluid phase to move around in. And so there's a limit to how much you can actually filter from that suspension. And that's been optimized. And then this holding tank sits here, and it has a valve here that opens occasionally. And hopefully you're prepared for that. And you know when it's full. That's a brain thing, when it's full. So let's take a look at inside the kidney at some of the anatomy. This is a slice through the kidney. And basically you can't tell a whole lot other than it has some different structures near the surface than it has in the internal. This is called the cortex. This internal structures are called the medulla. We won't need to deal with that. What's interesting is this. This is what was done here. You take an animal and you inject essentially like polyacrylamide into the blood of the animal, and it hardens. And then you dissolve away the tissue, and you're left with a total sort of picture, if you will, of the blood, arterial, artery, capillary system. So it looks like a tree. Except what you have to think about is all these capillaries out here then turn around and merge and come back out the renal vein. So now the other thing you might be asking yourselves is, well, okay, if this plasma is filtered, where does it go? Where does this 125 mLs a minute go to get processed? And it goes into something called the nephron. And you have essentially, let's see if I can take you through this. The blood comes in, here's the capillaries coming up, and you'll see it looks like grapes growing on the stalk here. It looks like these little grapes. These are filtering units. They're called glomeruli. GL, ER, ULI, or a single one's a glomerulus. And you have millions of these. They're all the same size. And the best way to explain it is the blood comes in, you've gotten to these little capillaries, and there's a little capillary tuft which has blood coming in and blood going out of the tuft. And imagine encapsulating that tuft with the blind end of another tube which leaves it. So that the stuff that's, the plasma that's filtered out of these capillaries gets captured, like in a funnel. Gets captured in the blind end of this tube, and this tube is the nephron, which processes that fluid. And what its job is to remove the solutes from the filtrate and then return what's left back into your circulation. And so you'll see that there's a capillary going in, a renal arterial going into the glomerulus, a venule coming out of the glomerulus. This gets back and gets pushed back into the renal vein. And so at the end of each of these capillary tufts is one of these glomeruli. And that's really the filtering unit. And these diagrams show it, I think, a little clearer. Let's take a look at this one up here. You see the blood coming in to this glomerulus, how it breaks up into a capillary bed called the glomerular capillaries. They all reform and they come out essentially a venule. And the filtration occurs across this capillary bed and gets picked up in this tube. And this is filtrate now, which is being processed in ways that allow the nitrogenous materials to be removed. The extra material is reabsorbed back into the capillaries, and this goes down into your ureter. And this gives you some idea of the capillaries that surround this tube. And the reason you need capillaries surrounding these tubes is they're picking up the material that's been processed and returning it back to the blood. So you actually have two capillary systems in parallel in the kidney. You have a filtering capillary system in the glomeruli, and you have a peritubular capillary system downstream, which is reabsorbing that extra material, this other 124 mLs a minute, and returning it back to the bloodstream. There's two kinds of glomeruli, nephrons, juxtamedullary nephrons, which are deeper in the kidney, and cortical nephrons, which are near the surface. We don't need to care about that right now. But this is just, I just do this to show you another picture. Here is the blood coming in. It goes into the glomerulus. It gets filtered into the tubules. And these tubules are also surrounded by capillaries, which are picking up the filtrate and returning it back. So this is a very cool system. Nature has designed a system which allows you to filter a lot of plasma from your blood, process it, and then return most of that filtrate back to your blood and shunt the solutes that you want to get rid of into these things called collecting ducts, then the ducts of Bellini, and finally into your bladder. And if you think about, who can do this calculation? 1 ml a minute translates into how many mLs in 24 hours? So 1 ml a minute is 60 an hour times 20 hours, 24 hours, is how much? About 1420, 1.4 liters a day produced. And how many liters a day are you of plasma is your kidney seeing? It's seeing 625 mLs a minute times 60 times 24. It's a huge amount, right? I mean, your kidneys process enormous amounts of fluid. So this is just another scope. This is the surface of the kidney. You can see that the blood comes in, it goes to the glomeruli, it gets filtered, it goes back in and goes back into the vein. And the little venules that come out of the glomeruli break up again into this other capillary system, the peritubular capillary system, which reabsorbs. So what you have in the kidney are two capillary systems in parallel, in series. You have the glomerular capillaries which filter, come back together, and then break up again into the peritubular system, which does the reabsorption, which is very interesting. Only capillary system like that in our body. And the reason I keep showing you these figures is maybe one of them will hit home, and you'll get an image in your head about how this works. So over here we have the blood in and blood out. We have the filtration occurring in this little tube. This is a blind-ended tube. The filtrate gets processed. You can see that these tubules are surrounded by the peritubular capillary system, whereas the glomerular capillary system is inside. Here's an example right here, right there. It's where the filtration takes place. Very intricate engineering design, if you want to think about it that way. Now, here's a fellow who draws anatomy and tries to make it look more real. And so here you see the capillary bed in here. And this is where the filtration takes place. The filtrate runs down these tubules. The hydrogen, creatinine, uric acid, they're all kept inside. The clean fluid goes back outside, gets picked up by the peritubular circulation and returned to your body. So what is it achieving? So in this table, here's your glomerular filtrate, 125 mLs a minute. These are the concentrations of these various solutes: sodium, potassium, calcium, magnesium chloride, bicarbonate, sulfate. Here's glucose, urea, uric acid, creatinine. To give you an idea, this is the concentrations in the urine. And this is the ratio of the concentration in urine to concentration in plasma. And look at this: urea 70, uric acid 14, creatinine 140. That shows you the concentrating power of the kidney for these various substances. It's enormous, and of course it has to be that way in order to perform its function. And this is the typical input of waste products in terms of grams daily input to the excretion pool. So you're talking 12 grams of urea in a day. I mean, that's a significant amount. Now, how do the capillary membranes work? Yeah.
Question.
Yeah.
What were the last three substances that were in the last table?
Those materials, I think, inulin was one of them. Inulin is a sugar, polymer sugar, that is actually used. I'll talk about it next time. It's not something that is normally in the filtrate, but because it's something you inject in somebody as a marker. I actually don't know what this one is, and this is para-amino hypochloric acid, or something like that, but it doesn't have any effect on what we're doing. These are used as markers in experiments, and inulin is the one I'll be talking about next time, show you how that's used. You'll see it on this graph, because this is what's key, and that is, what do the capillary membranes in your kidney hold back, and what do they pass through? So here's water. A radius from diffusion coefficient will normalize this to 1. Urea is a little bit bigger, about 1.6 times that of water. Glucose is about 3.6 times. Sucrose is about 4.4. See them getting bigger here? And look at filtrate to filtran. If there's a 1 here, what does that mean for these substances? They're not excluded. They pass through the capillary membranes unimpeded. But now what happened to inulin here? It's impeded just a little bit, 0.98. Most of it gets through. Now look at myoglobin. Now that's a molecular weight of 5,500. Look at myoglobin. Myoglobin is a protein, 17,000 molecular weight. If it gets in your blood, which it shouldn't be since it's supposed to be in your tissues, 75% of it can get through the capillary walls. Egg albumin, or just albumin, I don't know why you'd have egg albumin unless you ate it, but albumin, which you have, 43,000 molecular weight, 28 times the size of water in molecular diameter, although it's kind of shaped like a football, 22%. So most of it's held back. Hemoglobin, 68,000 molecular weight. This is the protein that's in your red cells. It's almost totally retained. And serum albumin, which is the albumin that's in your bloodstream, and which is a very important thing to keep in your bloodstream, 69,000 molecular weight, it's essentially all retained. So you can think of the capillaries as like a sieve, a molecular sieve. And anything that's the size of serum albumin or larger is held back. Obviously the cells, the white cells, the platelets, and the red cells are all held back. Everything else goes through. Now the kidney has to be sure that if in fact some sucrose got through or glucose got through, it wants to save those. So it has a means actually to pump those back in selectively and leave behind the urea and the creatinine and the uric acid. So it's a very interesting and a very precise filter. And now the question is now that you know all this physiology, how can you take that and fold it in with the data that I will tell you about next time and create a mathematical model using mass balances as to how the kidney processes blood and what are the controlling factors that are the determinants for how much filtrate you produce and how much urine you produce as a function of, say, blood flow to the kidney, pressures in the kidney, and so forth. So we'll learn about membrane ultrafiltration at the next lecture. And then I'll ask you, knowing all that, how would you design an artificial kidney to mimic all this? Think about it. It's pretty tough. You've got molecules going one direction, molecules going another direction, selectively removing some, not others, conserving the water. Big problem. But there are machines that do that, and you'll see that when we take the tour.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.